run the scene that light up your face. I run the place, I run the space, I run the scene. Run the place, run the space, I run the scene that light up your face. I run the place, I run the space, I run the scene.
And on this one is physics.org. Scientists solve mystery of DNA damage detection and repair caused by sunlight, alcohol, and pollution. A collaboration between researchers at the Laboratory of Medical Sciences in London and Laboratory of Molecular Biology in Cambridge has solved a decades-old mystery which could pave the way to better treat cancer in the future. The work uncovered the basic mechanism of how one of our most vital DNA repair systems recognizes DNA damage and initiates their repair, and that has eluded researchers for many years. Using cutting-edge imaging techniques to visualize how these DNA repair proteins move on a single molecule of DNA and electron microscopy to capture how they lock on to specific DNA structures. This research opens the way to more effective cancer treatment. The collaboration between the laboratory of Professor David Rudu and Dr. Laurie Passmore has been a brilliant example of how team science can bear fruitful results and underscores the importance of these two institutes in driving forward research that unlocks the fundamental mechanisms of biology, which will underpin the future translation of that work into improvement in human health. Unlocking fundamental mechanisms of biology. Can you believe we still don't have the fundamentals down? It's kind of amusing and sad and inspiring and frustrating. It's like everything all at once. There's so much to discover. You know, it's kind of like Star Trek when they're going out into the universe and they have so much to explore and discover and adventure. That's kind of how it is. We don't really know anything. So we have a long way to go before we figure everything out, if that's even possible. But the researchers were working on a DNA repair pathway known as the Fanconi anemia pathway, which was identified more than 20 years ago. It's going to be called FA after this. So DNA is constantly damaged throughout our lives by environmental factors including ultraviolet light from the sun, alcohol, smoking, pollution, and exposure to chemicals. One way in which DNA becomes damaged is when it's cross-linked, which stops it from being able to replicate and express genes normally. In order to replicate itself and to read and express the genes, the two strands of the DNA double helix first has to unzip into single strands. When DNA is cross-linked, the nucleotides, the steps in the double helix ladder of the DNA of the two strands become stuck together, preventing unzipping. And they have a video, which is linked in the article, and I'll link the article for anybody that wants to see the video of their microscopic beads. The accumulation of DNA damage include cross-linking, which can lead to cancer. The FA pathway is active throughout our lives and identifies these damages and repairs them in an ongoing process. Individuals who have mutations that make this pathway less effective are far more susceptible to cancers. Although the proteins involved in the FA pathway were discovered some time ago, a mystery remained over how they identified the cross-linked DNA and started the process of DNA repair. The team from the MRC LMS sister institution LMB in Cambridge, led by Laurie Passmore, had previously identified the D2I protein complex, which acts in one of the first steps of the FA pathway. Clamps onto the DNA, thereby initiating DNA repair at crosslinks. However, key questions remained. How does D2I recognize cross-linked DNA? And why is D2I complex also implicated in other types of DNA damage? The research published in Nature used a combination of cutting-edge scientific techniques to show that the D21 complex slides along the double-strand DNA, monitoring its integrity, and also elegantly visualize how it recognizes where to stop, allowing the proteins to move and lock together at that point to initiate DNA repair. So I've talked about DNA and protein being like a roller coaster, well, proteins being like a roller coaster. And if you think of this, for me at least it helps. If you think of this D2I protein sliding down the DNA, so it's basically like a specialized DNA or a specialized roller coaster track sliding down another roller coaster track. Or if you want to think of how the wheels of a roller coaster work, there's like the clamp that locks in on either side of it. This is kind of like that clamp running down the length of the track, and whenever it finds a spot that is broken, it stalls out and stops on that spot. So it's like riding the track of DNA. This protein, which is kind of also a track in its own way, but it's specialized to fit to do this and in its shape. So yeah, that's kind of trippy to think about, but I think that might help people imagine what's going on here with this thing clamping on, because it said that it was clamping onto stuff. So then we have Artur Kazmarski and Korak Ray and David Rudea's single molecule imaging group, working with Pablo Alcón and Lori Passmore's group, used a state-of-the-art microscopy technique known as correlated optical tweezers and fluorescence imaging to explore how D2I complex slides along a double-strand DNA molecule. Using optical tweezers, which is the crazy term even in itself, they could catch a single DNA molecule between two beads, which allow them to precisely manipulate the DNA and incubate it with chosen proteins. Using fluorescently labeled D2I and single molecule imaging, they observed how individual D2I complexes bind and slide along DNA, scanning the double helix. They discovered that rather than recognizing the cross-link between the two strands of DNA directly, the FA clamp instead stops sliding when it reaches a single-strand DNA gap, a region where one of the two strands of DNA is missing. This is just hard to imagine and wrap your mind around the complexity and the different types of interactions going on here, but DNA is a representation of information. I mean, it's like a physical representation of information. So it's talking about scanning the DNA. This is like a computer program that's scanning information for abnormalities or issues, degradation in the data. So if you think of this in a computer term, it's crazy how the computer and scientific electronic type devices parallel so well with the biology and mechanical types things, the biomechanical interactions, processes. It's fascinating that one could make those parallels. Using cryo-electron microscopy, a powerful technique which can visualize proteins at a molecular level, the researchers next determined the structures of the D2I complex both in its sliding position and stalled position at the junction between single-strand and double-strand DNA. This revealed that the contacts D2I makes with the single-strand double-strand DNA junction is distinct from contacts it makes with double-strand DNA alone. This allows them to identify a specific portion of the D2 protein called the KR helix that they showed in their single molecule imaging experiment. It's critical for recognizing and stalling at the single strand DNA gaps. So working with all these people, the scientists, they further showed that the D2I complex ability to stall at these junctions using the KR helix is critical for DNA repair by the FA pathway. So they identified this other protein that is also a key element. This is just like a bunch of proteins all working together in different ways. When DNA normally replicates in our cells, it unzips the two DNA strands and copies each single strand. This creates a replication fork where the original DNA strands are unwound and new DNA, double-stranded DNA, is formed on each strand. However, when this fork reaches a DNA crosslink, the strands cannot be unzipped, stalling the usual DNA replication process. This stalled replication fork thus contains exposed single-strand gaps where the DNA has been unwound but not replicated. This research has shown that it is these junctions between single and double-strand DNA at the stalled replication fork that the D2I protein complex latches tightly onto. Not only does this allow the D2I complex to bring other FA pathway proteins to the DNA crosslink to initiate repair, but it also anchors the remaining double-strand DNA, protecting the stalled replication fork from enzymes in the cell that would chew up the exposed end of the DNA strand and further damage the DNA. So this D2 complex, it's also like an alert to bring in more FA pathway proteins to help initiate the repair. So it's like an alert going out, and it also anchors itself to protect the replication forks. And so it's like a protective thing too. It's covering it all up like a band-aid. How is it that this stuff evolved to do this is a major question for me. Because it's almost like how, like we talked about earlier, of brain activity in dying patients. How does the brain evolve to do something as it's dying? If it's dying, then it never got the chance to pass on its information before it died. So how is that information being carried on? That's just a question that I have. I don't have an answer for it, but it's something that I see on a lot of different things. I ask myself, how did this evolve? And some things are more obvious than others, that's for sure, but this work has shown that it is DNA structures within the replication fork that stalls as a result of cross-linked DNA, rather than cross-linked DNA itself, that triggers the D2I complex to stop sliding and clamp onto the DNA to initiate repair. These stalled replication forks appear in many types of DNA damage, explaining the broad role of the D2I complex in other forms of DNA repair as well as via the FA pathway. Understanding the process of DNA repair, and importantly why it fails, holds huge importance, as DNA damage is a key factor in many diseases. Critically, many cancer drugs such as cisplatin, I don't know if I'm pronouncing that right, that doesn't sound right, working by inducing such serious cellular damage to cancer cells that they stop dividing and die. So it's notable that this cisplatin drug just increases the cellular damage to the point that they die. We don't have anything that will make the cells come together and like actually work correctly. We can't actually get the DNA and stuff to work correctly. All that we can do is make it worse. And that by making it so bad, that's how we kill cancer. I think that that's kind of notable, that we can't fix anything. All we can do is just make it worse, as far as this DNA thing where we're at right now. It's kind of interesting. But last paragraph here, last two sentences. In such cases, DNA repair pathways, such as a vital physiological process in normal life, can be hijacked by cancer cells who use them to resist the effects of chemotherapy drugs. Understanding the mechanistic basis of the first step in DNA repair pathway may lead to the ways of sensitizing patients so that cancer drugs can be more effective in the future. So I guess they're going to try to target certain cells specifically. So we'll move on here to something that I found absolutely fascinating. Another really good one from physics.org. And these are all from like the last 2 or 3 days, I think. The only one that wasn't was from like the 20th, the 24th. But these are all from like the 29th and 30th, 31st, that sort of area. But you don't need to read the news or scroll through Instagram for a long time to stumble across the latest example of a rare and beautiful species that has gone extinct. Since 1500 AD, at least 705 species of vertebrae have gone extinct and 571 species of plants have gone extinct. So the first farmers often made landscapes more biodiverse, providing lessons for rewilding today, which is fascinating. Probably one of the most fascinating things I've read in a while. Humans have now appropriated over half of the Earth's surface for farms in urban areas, and this is primarily to blame for these recent declines in global biodiversity. But humans didn't suddenly appear in the year 1500. Early humans were burning the African savannas from about 400,000 years ago and possibly earlier. There's evidence that Neanderthal altered the plants and landscapes of Europe about 125,000 years ago, and while there is some debate, humans were probably the decisive factor in the extinction of most of the Earth's once widespread megafauna over the past 100,000 years. I don't know about that, but it's interesting. Then, about 12,000 years ago, the most recent ice age ended and a new geological unit of time known as the Holocene began. This marked a major shift in human environmental relationships as people independently transitioned from foraging to farming many different places around the world. And I'll say super quickly that now, today, we are in the Anthropocene. And that's the die-off that we're in right now. And the reason we called it Anthropocene, anthro means human or human-like. So they're trying to say that it's a human-led extinction, but they're talking about renaming Anthropocene to make it more specific because humans aren't the ones that are destroying the Earth. It's a very small minority of the humans on Earth that are destroying the Earth. And those are like the ultra rich, the billionaires of the Earth are the ones that are destroying it. And these massive corporations, those are the ones destroying it. So the name needs to reflect that. So they're talking about renaming it to reflect something more in line of like a capitalist type of extinction event rather than a human-led extinction event. It's a capitalist profit motive-led extinction event, which is more to the point and succinct historically. So yeah, I'm behind that name change 100%. But whether this transition looked like rice paddies in China, wheat fields in the Levant, and maize and squash production of Mesoamerica, humans were modifying the landscape more and more. Domesticated livestock soon followed in many places. All this had surprising impact on biodiversity. Our latest research shows farming and other human-driven ecosystem changes increased diversity as often as they reduced it. Fossil pollen tells us about past vegetation. To reconstruct how vegetation has changed over many thousands of years, scientists look at fossilized pollen grains contained in layers of sediment and organic matter, such as decomposed twigs and leaves. In places like peat bogs and lakes, the timing of layers and variety of pollen found create a picture of the changing diversity of plant species through time and local landscapes. Analyzing pollen data from the Neotoma Paleoecological Database, we found that plant communities became increasingly diverse across most of northern hemisphere over the Holocene, starting around 9,000 years ago in Europe. Surprisingly, these increases in diversity are often linked to growing levels of human activity. In the southern hemisphere, the results are more varied. In Africa and South America, increased human land use saw decreases in plant diversity, and reduced land use saw an increase in diversity. Despite this, the turnover rate, how quickly different types of plants change, increased with human land use in all of the continents. Humans have been a key driver of vegetation change for a very long time. Farming created a patchy landscape. Following the end of the last ice age, the climate warmed, conditions generally became a bit wetter, and forests spread across newly thawed lands. Particularly in the northern hemisphere, it is in these expanded early Holocene forests where we find the strongest overall links between an increasing human presence and increased diversity of plants over the Holocene. In contrast, in open and grassy locations such as the North American Great Plains and savannas of Africa, plant diversity decreased as human pressures grew. The link is best illustrated in Europe, where two large increases in plant turnover and biodiversity found in pollen samples roughly coincided with two major migrations of people into Europe over the Holocene. The first began 9,000 years ago when the north and westward movement of the Neolithic farming populations from the Fertile Crescent region of the Middle East into Europe. The second began 5,000 years ago during the early Bronze Age as horse-riding pastoralists from Central Asian steppe, the Yamnaya, moved westward into Europe. We think that these new arrivals in Europe would have chopped down some, but not all, of the forest to make way for animals and crops to make their homesteads. Grasses and other plants that thrive in disturbed ground would have moved in. This would have created a diverse landscape full of patches of different vegetation. Making a forest patchy is easier than making an open grassland patchy, since it's easier to chop down trees and for open ground plants to colonize these gaps. Making open grassy location patchy means planting trees and them surviving in places that are too cold or too dry to support tree growth. We believe that the fact is it's easier to make forests patchy could have driven some of the historical differences and patterns of vegetation between different parts of the world. Again, relating this to South America and the Amazon jungle, of what we learned about. We won't even talk about the terra preta of how they biologically engineered the soil, but just talking about how we have discovered that the Amazon was manufactured, it was grown, and it's in a pattern, and it's in a, if you could say, like a checkerboard grid type of thing. That sounds like what they're describing right here. So that fits perfectly in line with the strategy of how the Amazon was manufactured. And if ancient early humans could create these utopian locations of the earth where there's just food everywhere and everything's so biodiverse, that means that we can do it again today. And even if we are losing mass amounts of life on the earth, we can still make areas of the earth that will have the biodiversity and grow, and that those areas will continue on into the future, perhaps, for eternity, or who knows, there might be another ice age and it keeps on going until the next one. And even like after the civilization as we know it is gone, but the jungles and the Amazon that they created are still there, which is the case of the Amazon today. That those civilizations are not necessarily intact the same way that they were in the past. I mean, those people are still there, right? That's who we're getting the information from, is the people of those places. But it's not the same as it was in the past. But we can still take the lessons from that. So, land without people may have less biodiversity. And this is all just theoretical, remember, as well. This is a theory. And it's very Eurocentric as well, this one article. So this is just these one scientist's take of it. It's not necessarily the authoritative truth. This is on all of this stuff. So yeah. Many people today advocate for enhancing biodiversity by minimizing the human influence on landscapes. Often this takes the form of rewilding, which aims to let nature lead. Yet our results suggest that in many places, though not all, minimally human-modified landscapes had fewer different types of plants. Often, human disturbances enhanced rather than eroded biodiversity. Indeed, many of Europe's most biodiverse places today are traditionally managed. Low-intensity farmland such as alpine meadows and the de Haces and Montados in Spain and Portugal. In light of this, removing humans from landscapes to make ecosystems healthier and more diverse could sometimes be counterproductive. Keyword sometimes. Though perhaps surprising, our research reveals that in many places, biodiversity thrives because of, not despite, thousands of years of human interaction with the Earth's ecosystem. So again, I'll just very quickly say it's because they were taking care of the planet and taking care of the land and the water and the plants rather than destroying it and just not caring about it. I mean, it's just two completely different ideologies and philosophy of life, of how they interact with the world around them. Either you can destroy it or you can nurture it and grow it. And I feel like that's what we're reading about here. They just didn't put it in those terms. But I'll end it quick here from Ohio. And there's a video if anybody wants to watch. But Ohio State Highway Patrol trooper stepped into action last week using a tasty treat to help rescue a dog that got loose. The dog, who had been loose for days, was located near a ledge on a railroad overpass near Ohio State Fairgrounds. The trooper took into action using potato chips to help rescue the dog and reunite her with her family. There's a dog on this track. I guess it's been missing for 2 days, the trooper could be hearing saying on the body camera. He walked along the side of the tracks until he found the dog near the ledge of the overpass. Eventually, he resorted to using chips he had on hand to lure the dog over so he can rescue her from the ledge of the overpass. So, got to give it up to the animal rescue and quick thinking of having some potato chips nearby. Who would have thought that, like, the people making those potato chips in the factory would have ever imagined that the chips they made that day would save a little dog's life? So it's kind of funny to think about. But, yep, I hope that you guys are safe out there, and I'm glad to be back, and I'll definitely put out some more stuff soon. Thank you so much for listening, and let me know what you learned or any of the disaster movies that come to your mind of what is going on today.